Thank you, Dr. Morris, for your kind introduction. Actually, that Dr. Jensen ended in a really great place because my talk's going to be on MRI, and he gave a great introduction on that, so that was really perfect timing. And in fact, we're also using a GE system, and while it was gone, they actually did the upgrade, so I'm really looking forward to all those good stuff that I'm supposed to get when I get back. Although I did get emails telling me that I have to go and make sure that my old sequences still work on the scanner. I want to acknowledge that this work is in collaboration with Dr. Tanya Adam. She's the neurobiologist in this collaboration, and a lot of the stuff that I'm talking about in obesity are hypotheses and methods that she has come up with over her PhD work and her postdoc work, and I really want to acknowledge that I'm representing her work here. So I'm going to talk a little bit about why we're doing this work. Obesity is a huge issue. About 1/3 of U.S. adults are obese, and it comes out to be about 10% of the entire healthcare costs at $147 billion. This was estimated in 2008. In 2011, I'm guessing that might have gone up. So there really is a big need to look at obesity. And the research community has really moved from obesity being a socioeconomic problem to really thinking about it as a disease state. And therefore, you'll see why that this change in sort of perception of obesity has really allowed me to do the work that I do here. And Tanya's specific goals are not only in obesity, but she specializes in childhood obesity. And the reason why childhood obesity is important and different from adult obesity is that in the last 30 years, the rate at which obesity has been increasing is especially extreme with children. And that's very disturbing. Children tend to have a pretty stable rate of growth, and there's pretty good stable graphs and charts about how their weight gain and their growth patterns and things like that. And there's a lot of concern that the obesity at a young age is really going to cause some serious problems in adulthood. And these problems are going to be more severe than people who get obese in their adulthood. So just to talk a little bit about the landscape that I'm really dealing with here. In obesity research, there really is a ton of different ways to look at it. There are molecular biologists who look at cellular mechanics within the adipose tissue. They look at the cell biology. There are geneticists who really look at the genetic factors that affect obesity. There are behavioral scientists and psychologists that look at behavioral aspects of it, environmental aspects, clinicians in endocrinology and other fields that look at clinical aspects of it, and clinical researchers that look at the whole disease overall and how it's progressing through society and through what populations and so on and so forth. So I want to emphasize that Tanya and my approach really here is emphasizing on the behavioral clinical and really the progression of the disease through the population. So it's a population study with clinical and behavioral data, and we're really adding a dimension of imaging to the work that we're doing. So I'm going to go right into the study that we did, and this is an illustration of just what the community is looking at in terms of how to integrate fMRI and other MRI modalities into their research. We recruited, this is a sort of pilot preliminary study. We had 13 obese pre-pubescent girls between the ages of 8 and 11. The reason why they were 8 to 11 is we wanted them to be pre-pubescent, but we needed them to be old enough to tolerate the MRI. Can I have a show of hands of who's been in an MRI here? So, quite a number of you. I've been in there quite a bit, both from my experience and my colleagues' experiments. It's not a pleasant experience. Especially for a young girl, it's very loud, it's very difficult, and the first girl we did literally cried for her mother the whole time when she was in there. So this was actually a very, very challenging protocol to implement. And we did this on as I said earlier, a GE 3 Tesla system, which provided great SNR for the work that we're doing. So the fMRI paradigm we did was we had the girls go into the MRI, and they looked at a number of stimuluses. The first one's a control. So here I've shown you some of the images that we use, which is a door, a bike and a hiking trail. Although some of my friends say the door might lead to a restaurant and might lead to food, I think that's pretty safe as a control. In terms of high-calorie foods, I have hamburgers and candy and mac and cheese, and I left out my favorite, which was the fried chicken, which seemed to be a common one, especially in Los Angeles. And then we had low-calorie foods, which was asparagus, salads, corn, and it's quite obvious that these girls have a sort of a very different view of what these foods are and how they like them. When we showed them some of the vegetables, they didn't know what they were sometimes, and they didn't really, there was no excitement towards asparagus, for example. And the results of the fMRI experiment really show that, and really localize in the brain where these kind of thoughts and these sort of behavioral changes are occurring. So for those of you who don't know about fMRI, I'm going to just go through really quickly. This is the dirty 2-minute sort of introduction to fMRI. Below here is how we built the reference function. You can see these boxcar responses. The top one corresponds to the control. The middle one is the high calorie, and the bottom one is the low calorie. And what we do is we take these boxcar reference functions and we convolve that with the hemodynamic response function. And it's called the bold hemodynamic response because it's blood oxygen level dependent. And I'm going to tell you why this is important at the end of the talk. But just keep in mind that this is actually a blood flow dependent method. I.e., we're not detecting directly neuroactivity. We're talking about blood flow changes relating to neuroactivity. So I had this great slide by Tom Nichols that I would like to use. Remember those boxcars and that convolution that we're doing? You can see that in the green line on the left. And what you do is you take a combination of those. So you saw the function, the control, the high and low calorie foods, and we do contrasts that are based on those different reference functions. And what you get for each subject is areas of the brain that have activated either, have high responses with high-calorie foods versus control, or low-calorie foods versus control, or high-calorie foods versus low-calorie foods. We do all these different combinations, and we put all these images into a common space so that we can compare it between subjects. So this is what the image is. Let me see if I can use the pointer right here. And you can see that in every subject, there's a difference in the level of activation. And this is going to be important in a little bit. And this is, I can kind of tell, he didn't state what kind of activity this was, but this is most likely a visual cortex sort of activity in the back of the brain. And you can see that all these average up to a very clean activation in the back of the brain right here. And so we had similar results. So we pulled all the subjects together, and we looked at high versus control, low versus control, high versus low, and we generated sort of a list of what these areas are, and all of them made sense. It seemed to be working. And you can take a look at it with, visually too. Here are the spots that were significant in terms of the comparisons. But a lot of this is already known, so we really wanted to take this one step further. And there has been people who have been talking about integrating clinical parameters in a very in more than just a simple way with fMRI imaging. And that's what we did. Remember these green bars here that we saw? You can see that each person has a different sort of response to the foods. Not only are they responding to food, but the degree of response can be measured with fMRI. So what we did was we correlated those degrees of response to clinical factors. So what we had with these subjects was fasting insulin and insulin sensitivity. Both of these are indicative of insulin resistance. So I'll just go through the first figure here, A. This is an area that we deemed to be part of the caudate. And these are the parameter estimates of the caudate. What these indicate is the degree of activation the subject has to high-calorie foods versus looking at a bike, the control foods. And what this means is that when the subjects have, and each of these dots represent a subject. So when the subjects have low fasting insulin, that means they are not doing well. They're not you should have nice, high amounts of fasting insulin so that you are able to deal with rushes of sugar in your body and being able to break that down. And these subjects don't, and you can see that they have very low sort of brain activity relating to presentation of high-calorie foods. So what that really means was that a lot of these subjects, because or due to their obesity, they're not getting as much brain activity with the same amount of stimulus. And they're all getting the same stimulus, which are these images of foods. And this similarly occurred in the insula. And when you look at insulin sensitivity, it's a negative correlation, because insulin sensitivity is basically the amount of insulin in your body after you inject amount of glucose in your body, and then you perfuse the body with a certain amount of insulin. And after 30 minutes, they take your blood glucose level, and you can see that those who can't process the insulin and have a lot left over tend to have very, very low levels of activation due to high-calorie foods. And similarly, you can see what happens when we do even more interesting comparison, which is high-calorie versus low-calorie foods. And here again, the difference between high and low-calorie food activation, not big. Okay, so I'm going to go on to conclusions. These initial results we got pretty recently, so it really takes us some more time to really digest what this all means in terms of the neurochemistry and the neurobiology of the results. But the initial hypothesis that Tanya made was a decreased brain reward really correlates with insulin resistance. And we use fasting insulin and insulin sensitivity as our indicators for insulin resistance. And as you saw, people who were, these children who were obese just didn't get the same activation in their brains when they looked at sugary foods and high-calorie foods compared to when they look at low-calorie foods. So normal individual, you see a hamburger, you're very satiated, you get satiation signals in your brain. These people don't. And that's very surprising, considering that when we talked to them, they were very attracted to high-calorie foods. You would think the opposite, which is that their brain would activate more because of what they like. But somehow it's different. This is very interesting, and this is really where I think the work really is to really figure out why this is so. And I hope that my presentation has sort of showed how fMRI can be a powerful tool in obesity research. And there are different combinations. I didn't really talk about the parameters I use for this fMRI experiment. There are people who use this in very different ways, different presentations. There are researchers I know that are trying to, for example, feed a tube into the MRI so they can have some milkshake before they look at the images and look at how that changes their brain responses, and really very creative ways of investigating the mind of an obese person and see what really happens to the brain. Because really, when you're obese, the whole paradigm now is that it's not because you want to eat. It's almost a need, and it's almost addiction, and it really is a disease process. And we should treat it so, and we should use the imaging tools that we have to research it in such a way. There is one sort of caveat to this, and that is the interpretation results is very important. And my personal difficulty in working in this field is that a lot of the researchers are used to simple tools like insulin sensitivity or fasting insulin levels or BMI or other behavioral tests, interest tests. Imaging data is very complicated, and a lot of the results are there are lots of confounds in the results, and you have to be very careful about the information that you handle. For example, I mentioned that hemodynamic response function was very important. You have to remember that hemodynamic response function is really a surrogate for neuroactivity. And this neuroactivity could be generated by receptor uptakes. It could be generated by different kind of electrical responses. And this is really an aggregate of all that activity, and we're subtracting that. And you can't really make direct influences on what is exactly going on. But you can tell that there is a difference and there's something interesting there. But you have to be careful about the interpretations that you make. personally, because I love using MRI, I think there needs to be more work correlating MRI data and clinical data, and this includes structural and functional. I talked a lot about sort of functional in fMRI. We did some functional work, but you can really use structural information too, through the structures in the brain change. With these obese children, do the volumes. That's the simplest thing, but you could really look at the morphology of the brain in many very interesting ways. And specifically with MRI, you can look at T1 and T2 contrast in these regions, in the different structures in the brain. Structural volume analysis, there's a software package called FreeSurfer that gets you cortical thicknesses and volumes of different structures in the brain. The other work of my colleague Bryce Wilkins in my lab, who works on functional connectivity, really talking about how the default mode networks changes in the brain. And finally, the other part of my research, which is DTI, I look at the white matter connections in the brain and how that changes with obesity. And I want to acknowledge my mentor and advisor, Dr. Singh, who couldn't be here today, but he has contributed significantly to this work and my funding sources as well. Thanks.
Okay, thank you. Have a question?
One question about how do these results compare to when you take images of non-obese girls?
Yeah, so that's a good question. Actually, one of the problems was this was a pilot grant and we couldn't have control subjects. But we did do some control subjects within the lab, and other people have done that before. And you would see, I think, that trend be similar. I think that trend that you saw, let me show you here. In this graph, I think those data would fit well into that, because really fasting insulin and insulin sensitivity is a much better sort of indicator of obesity than somebody who's large. And you can, and I think we experienced that with people we talk to. There are some people who are obese that would be rather large but are quite functional, athletic, and they do not have the same health problems as some other people. And that really has to do with the biochemistry of the body. And just because, and the research before has been using BMI or weight or any of these other indicators, and somehow that just didn't paint a full picture. And these indicators really are starting to show that it's more complex than just weight. I hope that answered your question.
Great, thank you.
How about the experiences related to the food group that you showed, whether it's high calorie or low calorie?
Sorry, could you repeat that again?
The food is, well, the experience that these obese girls had with the food that you were showing them maybe has any...
Oh yeah. So that's a huge behavioral aspect of it. And Tanya's actually, her initial training was in clinical psychology. So if I could show you, she's actually just received a K grant on this, and it specifically targets Hispanic population within the LA area. She tries to target populations within a similar neighborhood to control for those environmental factors and the cultural factors. They've shown that cultural factors affect the way that our brain responses to these different stimuluses. And really, we hope that this study, if it shows a significant difference from a general population study, then we know that these factors are really significant.
Okay, thank you very much.